So I just want to welcome you all. I'm Dr. Melinda Power. I'm an associate professor in the Department of Epidemiology in the School of Public Health. We've been chartered for about a year, so we're at the Institute for Brain Health and Dementia. We pull faculty from across the university, with most of our faculty coming from either the School of Public Health, the School of Medicine and Health Sciences, or the School of Nursing. So what do we do, right? What's our purpose? Our goal is to bring together faculty, students, staff who are interested in adult cognitive health. And in that context, we're looking to kind of grow our research base to really understand how we can better the lives of people who are either at risk of or experiencing cognitive loss. Right now, we have a number of different things that we do, on over time. One of those things is research and community outreach. So for example, we recently completed a brain health needs assessment for the District of Columbia Department of Health. And we have a community outreach committee that is trying to foster relationships throughout DC to try to see how we can do things like addressing disparities in brain health and to create partnerships to see what those communities really need around cognitive health. We also do a number of events. An example here is that our faculty members are currently working with the Alzheimer's Association to do a webinar about cultural competencies in dementia care. And we also have a lot of different opportunities for student membership. So we have hosted a number of practicum students, CE students. We have a number of volunteer research opportunities, and occasionally our faculty will also be recruiting paid research opportunities. When we think about brain health and dementia, we do cast a pretty wide net. The goal is adult cognitive health, but that includes everything from your typical Alzheimer's and dementia sort of classic presentation to brain health after TBI, to brain health in a number of other neurologic conditions, to just kind of general cognitive health overall. So I want to make sure we have enough time for our speakers, but if you have any questions at all, feel free to reach out to the brainhealth@gwu.edu email. I also encourage you to look at our website. We have a shiny new website, kind of covers who we are, what we do, and all of our different events and opportunities. So thanks to the Department of Epidemiology for letting us use this time today to introduce some of our faculty, and I'll turn it back over to Victoria to introduce our first speaker.
Great. So first up, we have Dr. Ciarleglio. If you want to go ahead and share your slides, you can go ahead and get started.
Yeah, so my name is Adam Ciarleglio. I'm an assistant professor of biostatistics in the Department of Biostatistics and Bioinformatics. And my time, so first I'll say I was gearing this more towards students. I'm trying to get students to do more research with me. So I'm going to talk about students, I think, a lot here. My time is split mostly between research and teaching. And so first, before getting into the specifics of some of the things that I do research on, I just want to talk about how I involve some students in that research. I have one doctoral student that I've been working with for the past year and a half, who's a doctoral student in the statistics department. And so we've been working together on developing some methods for handling missing data when those data are functional. So data that are measured over some continuum, whether it's time or space. And so we've particularly been looking at imaging data or neuroimaging data and relationships between neuroimaging data and certain clinical outcomes in depression, and how to handle missingness in that case. I also have been working with kind of one or two students at a time on CE or practicum projects. I have one student who's just finishing up her CE project, and she's done a really nice job looking at or doing a moderator analysis for a publicly available data set from the NIMH, looking at moderators of antidepressant treatment response. And so I'm always kind of keeping an eye out for students who want to work on those types of projects. And if you're interested in any of the stuff that I'm about to talk about, please reach out to me. On the teaching side, so I teach, I've taught the introductory statistics course a couple of times, but we have a lot of new courses now in our new department. And just this semester, we offered for the first time a class in applied categorical data analysis. And so we'll be offering that every spring kind of moving forward. And then next year, for the first time in the spring, we're going to offer a statistical and machine learning course that focuses on those methods applied in public health and biomedical research. And so I'll be teaching that in the spring. And I think we'll be doing that yearly moving forward. So if you have any interest and you have a good background in statistics and you're interested in those courses, please either reach out to me to talk more about them or sign up for them. Okay, so now let me get into some of my interests and current funding. So I'm currently funded on a career award from the NIMH where I'm developing statistical and machine learning methods for integrating clinical data and neuroimaging data to develop treatment decision rules for people with depression who need treatment for depression. So a lot of my interest is mostly focused on statistical methodology research, particularly in functional data analysis. Missing data problems was not something that I was originally interested in, but it's something that I've had to deal with some of the data sets that I've been working on, looking at this antidepressant treatment response. And then I'm also really interested in principled use of statistical and machine learning algorithms for prediction. On the applied side, I work a lot with collaborators in mental health. So I've done work in depression, schizophrenia, placebo response, and I really do enjoy that collaborative work. It's not necessarily work with developing statistical methods. So now let me talk about, I'm going to talk briefly about two projects that I've been working on for the past few years. One is a little bit older and one is newer that I've just started working on. I'll start with the older one. So I've been working a lot with trying to investigate biomarkers for antidepressant treatment response. And this is specifically what my grant is on. And so what's the problem here? So first I'll start out with what's major depressive disorder. It's a disorder that's one of the leading causes of disability worldwide. It has really negative consequences on a person's life, diminished quality of life, increased risk of suicide, lack of productivity, really terrible things. And so there have been lots of treatments that have been developed for depression, whether those treatments are actual drugs, antidepressants, or things like psychotherapy, or combinations of the two. But even though there's lots of treatments that are available, it turns out that the way that the treatments get prescribed, it's done in a really trial and error type of way because there's no really solid understanding of who's going to respond best to what treatment. There's tons of heterogeneity in treatment response. A clinician can have two people in front of her, both with major depressive disorder, and could give them both the exact same antidepressant treatment. One might respond really well, and the other one might not respond at all, or might even get worse. And so if that's the case, usually what happens is that subject who didn't do well will kind of find out after a few weeks that they're not doing well on the treatment. And then the clinician will try a different treatment for another 8 weeks. If that doesn't work, that process just kind of keeps going on until hopefully a decent treatment is found, but sometimes a decent treatment is not found. There's really terrible statistics associated with good outcomes, like fewer than 40% of patients with major depressive disorder show remission, like go from a depressed state to non-depressed state after their first 12 weeks on a particular treatment. And then even if you go through a whole round, a year's worth of trial and error, about 30 to 40% of those subjects are still going to fail to find a treatment that works for them. And so there's this push to kind of do a more objective, or have a more objective approach to kind of finding a treatment that's suitable for a person. And that approach is kind of the push has been to use biomarkers, or search for combinations of biomarkers that can be used to kind of guide treatment selection. And so I've been involved with a clinical trial that's known as the EMBARC Study. EMBARC stands for Establishing Moderators and Biosignatures of Antidepressant Response for Clinical Care. It's a multi-center RCT. It's got 2 stages, but we've mostly been focusing on the first stage, the acute stage, where we've got about 300 subjects with depression. They're randomized to either placebo or an active SSRI called sertraline, and then they're followed for 8 weeks to see how they're going to respond. The really cool, novel, amazing thing about this trial is that tons of baseline information are collected on these 300 subjects prior to their randomization. So typical clinical and demographic stuff that usually gets collected, like, what type of depression do you have? Is it anxious depression? Do you have insomnia? And, gender, age, all that kind of stuff is collected. But what makes this trial really cool is that in addition to those clinical and demographic characteristics, these subjects went through 2 days of neuroimaging scanning. So they would sit in the MRI for a while and get their structural information about their brain would be recorded. DTI, or diffusion tensor imaging data, we have for these subjects. They'd sit in the MRI and do tasks or just rest. So we have resting state and task-based fMRI data. We have EEG data. So all this huge amount of high dimensional data, we're trying to come up with ways to integrate it or investigate if any combinations of those data give us some information about how a treatment is, or how a subject is going to respond to treatment. So this is what I've been working on, is kind of the statistical side of this, is how do we take all of these really high dimensional multimodal data and kind of combine them in a principled way that could be useful. So we want to be able to identify moderators of antidepressant treatment effect. Because so here in this little picture that I've got, we're assuming that treatment has some effect on the outcome, but the kind of effect or the strength of that effect is likely to be, or the thought is it's likely to be moderated by clinical and demographic characteristics, structural information that's collected from MRI, DTI, and then functional information. And so what we're trying to do is to first identify kind of the relevant moderators, and then combine those moderators into a rule that would look something like this at the bottom. So it's a rule D that takes all of your data and then is able to say, well, if you have this kind of data, you should get treatment A versus treatment B, whether that treatment is sertraline or if that's a cognitive behavioral therapy or whatever. And so all of my research has been focusing on so developing these types of treatment decision rules, which will tell you, get treatment A or treatment B if some condition on this other function of the data, which I'm referring to as G, is satisfied. And so this coming up with this function G is the really challenging thing, because you have this kind of wide-ranging, high-dimensional data. And so the first few kind of approaches that we've been trying have been more along the kind of traditional functional data analytic methods. And so we have some papers that we published for some kind of new methods on integrating the different imaging and clinical data to come up with rules like this. And now we're kind of working on using more of the machine learning approaches like random forests or ensemble machine learners to kind of develop these G functions. So that's in a nutshell the kind of statistical research that I'm doing with this kind of antidepressant treatment response. The newer projects that I've started to work on recently is looking at depression in older or late life depression in frail subjects, and looking at the relationship between frailty and depression response or antidepressant treatment response, and just general kind of measures around depression. So I work with a collaborator at the New York State Psychiatric Institute in Manhattan. So we've done a couple of projects looking at this late life depression, and we see that late life depression is a chronic and heterogeneous disorder. And it seems like physical frailty is associated with greater severity of depression in older adults. And we've done some work investigating how frail subjects respond to antidepressant treatment. And it turns out that, what we've seen is that these frail depressed subjects tend to respond less to antidepressant treatment. And so there's this push to better understand what exactly is it about the frailty component that's making them not respond to antidepressant treatment. And then we're thinking that, if we have some kind of a way to kind of target this frailty, could we potentially make these people make their frailty kind of better so that they can respond to antidepressant treatment? And so there's all these issues around measuring frailty that we're starting to get into, kind of distinguishing between physical frailty and physical fatigue and mental fatigue. And so we have some really cool data where we're looking at, we have subjects, older subjects, with late life depression, who are kind of subjected to a battery of physical and cognitive tests. And we're looking at these data and seeing how they're related, and trying to just get a better understanding of what fatigue is in that context. So that's kind of ongoing research, and I'm hoping to involve some students in that here. So I think that's all I have to say. Thank you.
Great, thank you. Dr. Frame, it is your turn now to present.
Okay. So I am going to talk a little about the gut microbiome and nutrition, and I'm going to try and relate it to brain health, just to give us a kind of a teaser of what there is out there to be done, because there's a lot to be done. This is a definite gap in the literature, so I'm hoping you will all start incorporating this in your research. So think about that as we move forward. Also, I'm really active on social media, and you can follow me on Twitter or Instagram at PhD_Leigh. All right. Okay, so why do we care about diet in the gut microbiome? So diet actually has been estimated to govern the microbiota almost 5 times more than genetics. So our genetics is far less important than what we're putting inside of us, our diet. And what we know about diet, the microbiome, is that short-term dietary changes are typically transit. So you have a healthy diet and you go on a weekend binge of fast food and you come back to your healthy diet the next week. No harm to your gut microbiome has been done permanently. It will bounce right back. However, long-term diet changes can actually change the gut microbiome composition permanently. And that's the type of changes we're really looking at. So if someone moves from a fast food diet to a healthy diet after a period of time, say 6 months to a year, the permanent changes in their microbiome have been done. And hopefully in this case, it'll be good. The one thing we do know about diet, the microbiome, and how to define a healthy microbiome, which is not really definable at this point, but diversity seems to be the most important thing. So a diverse diet leads to a more diverse microbiome, and a more diverse microbiome seems to be what is healthy. So that is what we're looking for, and something that we can pin down in terms of the microbiome. Just looking at diet diversity may be enough to give us a lot of information about gut health. But what we typically talk about, we talk about the microbiome and diet, is fiber. And why do we talk about fiber? So fiber is digested by us in this upper part of nutrition where we typically talk about nutrition, but we get to this lower part of the GI tract, that's where it's being digested by the microbiome. And most fiber actually is indigestible to us, so it's actually mostly reaching the microbiome. And then they're going to process that, and it's actually going to contribute to our energy requirements. So up to 10% of the energy that we're utilizing from our diet is actually coming from our microbiome processing the fiber, not us processing it. And while they're doing that, they're producing short-chain fatty acids like butyrate, acetate, and propionate. Oh, I'm gonna go back. So about short-chain fatty acids, that seems to be one of the major indicators of the health beneficial properties of the microbiome. It's the mechanism, the reason that fiber seems to be so great for our microbiome is the short-chain fatty acids. And it's actually fuel for the colonocytes. So the colonocytes eat short-chain fatty acid, and they feed off of that, and that causes them to be healthy. So if we don't have enough fiber in the diet, we don't have enough short-chain fatty acids, the colonocytes can literally starve. And that can lead to poor barrier function, or as some people term that, leaky gut. What else is important about nutrition in the microbiome? So the microbiome actually produces nutrients. So we put food in and we give it nutrients, but then it also gives us other nutrients. And the nutrients that I have listed here are ones where the body stores, the pools that we have in our body are actually higher than we would predict from the diet. So if you look at how much we're taking in, that is much lower than what we see in the blood stores, which is kind of blows your mind. And where is it coming from? It's coming from the microbiome actually producing these B vitamins and vitamin K2. What else about the diet matters? So we've talked about micronutrients, and now we're going to talk about what else is in there. There's something called non-nutritive bioactives. That's basically everything else. So you got polyphenols, you've got all the phytonutrients, the phytochemicals that come from plants. And we know there's a lot of health benefits to these. Red wine, berries, fruit. We all know these are good for our heart and things like that. And why is that? It's actually that those chemicals are making it to the microbiome. About 90% of them actually go directly to the microbiome. We don't process them at all. And then the microbiome processes them. So without the microbiome, we wouldn't be able to process these compounds, which means we wouldn't get the health benefits from eating them. So in this case, taking in, say, red wine or berries, if your microbiome isn't able to properly process it, despite the fact that you're intaking them and you should see these health benefits, you may not actually see the health benefits if your microbiome is not able to fully process them. So I think that's a great indication as to where just looking at diet alone may not be enough. So why do we care about this with brain health? Because the gut is completely separate, right? Well, that's how we used to think about things, but it's definitely not the case. The whole human body is connected, and we can't really separate them. But it's particularly true in the gut microbiota brain axis. So that's sort of the fancy name we're giving for this relationship between the brain and the gut. And central to that is the vagal nerve. Which I'm sure all of you are familiar with, but many people don't realize that. They don't know that there's actually a nerve going from their brain to the gut, and it's directly talking to the brain. And what's happening in the gut is sending signals, and a lot of these microbes in here actually produce neurotransmitters. So these neurotransmitters like serotonin are being produced down here, and it causes a relationship with the brain. It causes it to do certain things. And how does that turn into disease. So you've got various different gut microbes, and I'm going to say, let's not focus too much on who is here, because that is a whole talk unto itself. But what we see is they have various different effects. So L. reuteri is in the gut, and it's producing whatever it happens to be producing here. And what happens is that leads to an increase in oxytocin in the vagal nerve, which goes on through the nerve and actually regulates neuronal plasticity. So that kind of sounds crazy, right? You've got a bacteria in your gut that is causing your brain to make new neurons or have them put out new connections. But that is what we are finding. This is actually from a Nature Reviews Microbiology publication. So this is not like some sort of edge fringe information. And this is true for a number of other products. And here we have short chain fatty acids again. So despite the fact that the gut is somewhat disparate and typically not included when we're talking about mental health, it's actually really important for mental health. And I would argue that it's actually foundational for all health. So looking at the gut microbiome and looking at the diet are important in any terms of research you're doing. So what my ploy for you is to think about this when you're thinking about designing your own research study. Are you incorporating the gut microbiome? Are you collecting information on the diet? Now, I understand everyone doesn't have all the money in the world. The gut microbiome can be expensive, but one of the nice things is you can bank it. You can collect stool samples and keep them at a -80 for, it seems almost indefinite at this point. So I would recommend doing that while you're doing in your study. You already have access to patients and you're collecting information. Try and bank stool. The other thing you can do is collect information on diet, and that can actually be pretty much free. Now, you might have to compensate your participants more because they're going to spend time doing this, but it costs almost nothing to collect information on diet. In fact, there are a lot of free apps out there like MyFitnessPal that you can collect this information, and now you have it. You don't necessarily have to know what to do with it either, right? So you could just bank this away as information. Of course, I could help you with that, but it may be that you're saving this until you've got your gut microbiome data, and this is just saving it away for a rainy day. I highly recommend that you take that type of approach because this is the future of science, and it is very difficult to study at this point, but one day it's going to be considered just run of the mill. So you might as well start dipping your toe into it. I also want to make you aware of some resources that we have. So we have the GW Microbiome Research Interest Group, which I founded and I am the chair of. And we have a listserv, which allows us to communicate with each other. And you can all get on the listserv by emailing me at leighrame.gw.edu. And we have a quarterly newsletter where I publish any publications coming out of GW related to the microbiome, as well as any really impactful publications that are really relevant to a large swath of the microbiome group. We've also talked about doing a journal club and maybe having some other meetings. So that's still in the works, a little bit on hold because of COVID. We haven't been doing any meeting in person, but that may change. The other resource we have is we actually do have a core lab here, and a lot of people probably don't know that, but we have a microbiome core lab, and that falls under Keith Crandall's group. And in our quarterly newsletter, we have a link to the core lab and also an article that they recommend as a really kind of a foundational basis on how to do microbiome research. So that's something I can send on to you to look at, and if you are interested in getting into this yourself. Of course, I can consult with you. Keith Crandall will consult with you too. The whole core lab will consult with you for no fee at all. They will just talk to you as colleagues. The other thing is you can follow me on social media because I do post a lot about this. So that might be a great way to get information and keep up with this because it is an ever evolving field. In fact, I'm sure what we learned yesterday and today will keep driving this into a whole new direction. Final plug. We have the GW Integrative Medicine podcast where we talk about a lot of things related to this. We talk about things related to dementia and healthy aging and things that I think will generally be of interest to you. You can get it anywhere you get your podcasts. I've got our main platforms listed here and I can take questions.
Great, thank you so much. We have a few minutes for questions if anyone had any of those.
And while you're gathering your questions, just know that you can please feel free to visit our website or email the brainhealth@gw.edu email if you have any questions for our institute or for our panelists today, and we'll be happy to connect you to whoever you'd like. So I'll leave it to the questions, and thank you all for attending.
maybe to get us started, I know a few of you are looking for student involvement. How might a student engage with this work if they were interested?
So the first thing I would say is if you're interested in any of the speakers that you've seen today, feel free to reach out to them directly and they can talk about what sorts of opportunities they might have for you to engage. If you're not ready for that step, you can go ahead and reach out to the brainhealth@gw.edu email address. We'll ask you to provide some information about what are your interests, what are the types of opportunities you're looking for, and then we'll do your best to connect you to Institute faculty that may have opportunities for you. So just feel free to reach out either through the Institute website or to the faculty directly, and we'll be happy to connect you, and hopefully you'll be able to make a good connection that way.